Hi everyone. Today we have amazing guest and author and congratulations of Editor's Choice article, Dr. Ashina, and Journal of Cephalalgia had excellent article we're going to discuss today with Dr. Ashina. And I am in San Diego, US, and Dr. Ashina in Denmark, connected by International Headache Society. So, hi, and please, Dr. Ashina, tell us a little bit about this excellent article and what brought you to this study, what methods did you use and design?
First of all, Nina, thank you very much for your kind introduction. And I'd like to thank also Cephalalgia and Editor-in-Chief for selecting our article for this interview. Well, this study is on non-vascular ATP-sensitive potassium channel activation in context to migraine. And the study was performed as a randomized clinical trial. Uh, just to give you a background about the study, why we decided to investigate this particular selective KTP channel opener in context to migraine. Well, as we all know in the clinical world and also in the research, that the migraine is a very common disease. It affects a lot of people, and we also know that one of the key features of migraine is that it can be triggered, provoked by the various triggers. And one of the common questions that the patients, usually people with migraine, ask us is about can we study those triggers? Because if we understand the triggers, maybe we can also understand the disease mechanisms. Well, over the many years in my lab, we conducted research focusing on experimental models. So using this, provocable nature of migraine, we used various molecules, natural occurring molecules, to trigger migraine attacks in order to identify those specific molecules or the specific signaling pathways involved in the genesis of a migraine attack. And what we discovered over the many years that all the triggers are vasoactive molecules, meaning that they also have vascular effect. They dilate, in particular, cerebral vessels, intracerebral, but some of them are also extracerebral vessels. And in this case, we can provoke attacks. Uh, how important is this initial vasodilating effect to trigger the chain of the events, eventually leading to a migraine attack? Still is a matter of a debate, and many people agree that uh, this effect is important, but some people say maybe this vascular effect is not important. It's kind of a B effect and not really important to initiate a migraine attack. But so far, we haven't studied a non-vascular molecule which was able to trigger the attacks. So one of the major discoveries coming from my lab was that when we showed that adenosine 5 triphosphate sensitive potassium channels are involved in migraine pathophysiology. Uh, those channels are widely expressed throughout the trigeminal vascular system, which we know that it is an anatomical and physiological substrate of a migraine. And we showed that by infusing one of these channel openers called levchromakalim, we are able to induce prolonged dilation of cranial arteries in healthy participants with no history of migraine. And we can also trigger migraine attacks in individuals with migraine. So these findings, position this channel as a potential and novel target for migraine treatment. But again, going back to this uh, mm, idea that it could be just a channel opening causing the migraine attacks and the vascular effect it's a epiphenomena, we searched for one of the channel openers which is non-vascular, meaning that this selective opener of KTP channel, which we know that it is preferentially targeting these channels, opening these channels in neuronal and beta pancreatic cells, but not those which are located expressed in the vascular smooth muscle cells. So we decided to conduct a study using this selective opener called NN414. And our hypothesis was that it would neither trigger migraine attacks or cause significant changes in the cerebral hemodynamics. And hemodynamics, we call it the dilation of the vessels or other changes in the vital parameters such as the blood pressure and heart rate. And to test this hypothesis, so what we did is that we conducted a randomized and double-blind placebo-controlled studies, which included individuals with migraine without aura. So, and the method was pretty straightforward. As I mentioned, that we did it in the randomized double-blind fashion, and we included also measurements of the diameter of the frontal branch of the superficial temporal artery to show whether there is any dilation, yes or no, because we know with the vascular KTP channel openers, we see a nice dilation. And we also included recordings of the middle cerebral artery velocity, so we can actually measure the velocity in the deep brain arteries using transcranial Doppler. So all this data was collected, and an analysis included the patients who completed the study. And in this study, we had 12 individuals who completed both study days, and they were included in the final analysis. So the findings are very interesting, and the main finding was that this particular KTP channel opener, NN414, did not cause headache or migraine attacks in individuals with migraine without aura. And also importantly, that this drug did not alter the cerebral and systemic hemodynamics, meaning that no vascular effect. Now, you might ask me, what's the difference between levucromocalim and this NN414? As I mentioned before, it is it is opener of the channels in neuronal tissue, in the neuronal cells, and also in the pancreatic beta cells. So the just to give you also idea about these channels, they're quite interesting topic because they're composed of the composition of these channels is that the various elements of these channels, which we call it the inward rectifier potassium KIR, and there is also other regulatory receptors, sulfonylurea in this context, and the various of these KIRs and SURS subunits composition, they produce a different channels, which in fact that feature different morphologies, functionalities, and also they have a unique tissue expression. So it depends which composition you have, and this composition is expressed in the specific tissues. Okay, so this one, NN414, it is it is a selective opener of one particular composition called KIR62 slash SUR1 subtype KTP channel. And as I said, it's primarily expressed in neurons and beta pancreatic cells. So when you give this drug, usually you get increase in blood glucose levels because it affects our insulin production. So that's why we had a control also in our study where we measure the blood glucose to make sure that in fact that we are giving something active, and we saw the increase of blood glucose levels at certain time points, which corresponded to pharmacokinetics of this drug. So and it's also rapidly absorbed after the oral intake. So it was administered orally. So these channels, as I said, are expressed in the neuronal tissue, and we were expecting that they would not induce attacks. In fact, that there are some preclinical studies that they suggested that administration of this drug will, in fact, have probably anti-nociceptive effect, because if you give something that you know that causing nociception, such as bradykinin, or you stimulate by thermal stimulation or mechanical stimulation, uh, in fact that this drug mitigated, or the opening of this channel mitigated actually this effect. So in fact that I wouldn't say it's surprising. It is confirming the hypothesis that the pure, or the channel opener, which opens the channels in the neuronal in neurons, wouldn't have a pro-migraine effect, causing the migraine attacks. And going back to our early studies that we show that levrochromacalin inducing, it is interesting because levrochromacalin is an opener with a high affinity to another composition channel subtype, which is primarily expressed in the vascular smooth muscle cells, and particularly cranial arteries. So again, uh, underscoring the importance of the vascular KCP channels, which we know that that leads to the efflux of potassium, hyperpolarization, and vasodilation, causing migraine attacks. So that's why we think with this negative study in terms of the outcome, no migraine triggering effect, we reinforce the idea that the opening of the vascular channels are important in the genesis of migraine attack. And we suggest that development of the selective composition of so-called KIR61 and SUR2B antagonist, they might represent a novel strategy, for treating migraine, whether it will be for the acute attacks or for the prevention, the future will show. So, but basically, we just say right now that these channels are important in the genesis of migraine attack, and the future will show whether the drugs acting on those channels, the specific drugs, are efficacious in migraine. So, and yeah, that was a kind of a short version of the study. And I hope that the future will bring more results. And we are working in this field, so we don't give up. And we hope that there will be also interest from the industry to develop the specific, the selective antagonist for these channels expressed in the cerebral vasculature.
This is spectacular and gives so much inspiration and hope to all of us for new therapeutics in the future. Thank you so much for this excellent work. And we will be looking forward for more articles with Cephalalgia and International Headache Society with excellent new results. So thank you so much, Dr. Ashina.
Thank you so much, Nina, and for this invitation and giving me an opportunity to present our study. Thank you.